Bonjour à toutes et à tous. Je suis très heureux de vous accueillir pour cette 12e journée scientifique du CRCHUM, pour la deuxième et j'espère dernière fois en mode virtuel. Je vais remercier tous les conférenciers qui sont dans la salle. C'est un peu curieux parce que je vois, on a un grand amphithéâtre, j'ai quelques conférenciers avec nous, mais évidemment je ne vous vois pas, vous tous qui êtes connectés, et je vous remercie de vous être connectés. Nous avons un très beau programme pour ces deux journées autour des forces cohésives de l'atome au traitement. On sait tous qu'on a vécu dans les 2 dernières années quelques forces non cohésives, ou je ne sais pas quel est l'opposé de cohésive, mais disons disruptives en anglais, et que notre communauté a fait preuve de beaucoup de forces cohésives. Et aujourd'hui, on les célèbre tous ensemble en attendant de pouvoir se retrouver pour le faire en personne. Alors vous le savez, nous avons un très beau panel de conférenciers. La matinée se terminera par la conférence d'honneur de la professeure Fabiola Gianotti du CERN. Et je voudrais vraiment remercier les membres du comité d'organisation de ces 2 demi-journées, Ouda Baïg, Manon Chouanière, François Deblois, Sabri Ahmed Rayal, Linda Adam, Mireille Chalifour, toute l'équipe technique, Chanel, Anne, et tous les gens qui nous aident à vous présenter cet événement. I'd like to say a few words in English to welcome those of you who are joining us from far, far, far away. I hear there are people connected from as far as New Zealand. That's great. The CRCHUM has connections everywhere in the world. So welcome to you for these 2 days. Thank you for staying up very, very late or waking up very early. I don't know. It's the middle of the night where you are, and we're certainly welcoming you with us today. Vous aurez noté que ce matin, nous avons une première session qui est quand même assez exclusivement masculine. Ce n'est pas un choix, c'est juste que dans la thématique de l'imagerie spectrale, je ne sais pas, il doit y avoir un intérêt pour cette thématique qui est liée au chromosome Y ou quelque chose, mais ça a été très difficile de trouver des conférencières, mais vous verrez qu'au fur et à mesure de la journée, et surtout demain, l'équilibre des genres se rétablit dans notre programmation. Donc, merci en tout cas à tous les conférenciers d'avoir accepté de venir ce matin. Et sans plus tarder, je laisse la parole à notre premier modérateur, François Deblois. Merci François d'être avec nous et je te laisse aller.
Merci beaucoup, Dr. Poitout. Donc, j'ai l'honneur d'ouvrir cette première session ce matin en vous présentant un collègue, ami, professeur Bouchard. Donc, professeur Bouchard a obtenu sa maîtrise en physique médicale à McGill en 2003. À partir de ce moment-là, il a commencé à travailler au CHUM. Il a, entre-temps, complété un doctorat aussi à l'Université de Montréal en 2010. Il est maintenant professeur agrégé en physique à l'Université de Montréal et il est aussi chercheur régulier au CRCHUM. Donc, sans plus tarder, je présente Hugo, qui va nous présenter les applications émergentes de la tomographie spectrale.
Merci beaucoup, François. J'aimerais remercier le CRCHUM pour cette invitation et je vais donner ma présentation en anglais. So, I'm going to talk about the emerging applications of spectral CT. I am a trained radiotherapy physicist, and for me it is very natural to start this talk with the following slide. So this slide summarizes the treatment planning process. So when a patient gets diagnosed with cancer and radiotherapy is the option for treatment, we follow a series of steps from which starts the CT scan of the patient. From the CT scan, we will contour the targets and also the organs at risk. That will, in order to treat the target, but also limit the toxicity to the organs at risk. We further proceed to a series of physical modeling where we do tissue characterization. So we characterize what type of tissue, the elemental composition, and mass density, and also the modeling of the beam transport in the patient. Once we have all this model in control, we can optimize the treatment so we can choose the configuration of beams that will provide the optimal dose distribution that will fulfill our objectives. So maximizing dose to the targets to control the tumor and also limit the toxicity to the patient. So CT has three very important roles in radiotherapy. First, it's positioning, so meaning that we want to represent accurately the geometry of the patient at the time of the planning. We also want to identify the targets and organs at risk, and we also want to quantify important parameters that are used for dose calculation. For the ones of you who are a bit less familiar with computed tomography, let me introduce you to the principles. So CT is a tomography of X-rays. So we basically have a detector attached to a source attached with a detector system, and that apparatus will rotate around the scanning object. And we do a projection, so we do a series of radiography around the scanning object, and we acquire a so-called sinogram. This is the image that you see in the middle. And using radon theory, which was established about 100 years ago, we can reconstruct the object in the real space. So leaving a couple of details aside, this technique is very well used, very well understood, and also available in the clinic for many decades. Now, please notice that all the images I'm showing here are black and white, meaning that defining a window level, you will go from that would go from 0 to 100%, for instance. You would assign 0 to black and white to 100, and then any level in between would be gray levels. And so meaning that there is only one channel of information per pixel. So what is different in spectral CT is basically you have many channels. So I'm representing these channels with colors. So spectral CT is a colored CT, except that these colors cannot be seen with the human eye. They are energies. They're not necessarily three channels. You can have an arbitrary number of energies. And what's interesting is that you can get complementary information from one image to the other. So once you acquire the polychromatic sinogram, you decompose this sinogram into all of its channels, and you can either reconstruct each of these channels individually, or you could do a couple of tricks and get the sinograms to have a conversation together and give you sinograms of physical parameters. There's also a few technologies that is available clinically. So I'm including here dual source or dual energy CT, which is of course more than one energy, so that fulfills the definition of spectral CT. So we start with dual source CT. This technology was developed by Siemens, I think around 2008. We, in the radiotherapy department, we acquired one in 2013. And essentially it is two CT into one. So we have orthogonal sources that acquire sinograms at two different energies simultaneously, so that we don't have any motion artifacts, or and we can reconstruct these images independently. Rapid KV switching is also a technology that was developed by GE, and essentially the idea is to alternate the voltage used to accelerate the electrons and create your Bremsstrahlung spectrum. So you get two energies alternatively that oscillate with time. And while you do that, you will also acquire alternatively sinograms of different energies. A third technique is the so-called dual layer detector developed by Philips, and what they basically do is they use a filter at the detector level, and they have two layers of detection, so they can discriminate the energy. Now, at this point, we're a bit more interested in arbitrary number of energies. So if we want to go beyond 2 energies, we need to use the photon counting technology. I'm giving you 2 examples here. We have the Siemens photon counting prototype. This image is taken out of the website, and also the CERN technology Medipix that is available in the Mars scanner. And how it works is basically every time there's a photon that creates interactions in a detector, you get a cluster of charges, and the pixelate anode will collect these charges, and the amount of charge is proportional to the amount of energy deposited. So you can measure the energy for every single photon that interacts with your detector, and you will essentially bin your counting in the same way you would do with the pulse height spectrum. Okay, so we started doing research about 9 years ago at CHUM's radiotherapy department, Department of Radiation Oncology. And I'm very proud to say that we were the first radiotherapy center in North America to acquire a dual source CT that would be dedicated for radiotherapy, and also 3rd in the world, I think, after Dresden and Heidelberg in Germany. And we started. Back then, I was a young scientist, and my boss had asked me to starting investigating the use of this technology, and we initially started in 2012, and quite rapidly we built a team, which you can see on the right, all my colleagues involved in this research. And I'm just going to summarize a little bit what we have done, or at least my contribution to this bigger team. Before I start talking about research, I really want to show you this slide. I like to answer this question: why is research useful? And the answer is in translational research. Why do we do research? We want the research to be useful. And what is the translational research model? Is going from the great questions of life to quality of life, happiness. You know, everybody likes happiness, I hope. And what I mean by great questions of life is, how big is the universe? What is matter made of? What is dark matter? Do we understand how the subatomic particles interact? And all these questions involve very, very expensive research, very big labs, and a lot of technology, and a lot of very bright scientists. And in the end, all the developments that are necessary to answer these questions can have a use that was not the initial goal of that technology. I'm giving you an example here for the Medipix that was originally a particle tracker for the Large Hadron Collider at CERN. But now we realize that this can be very useful for medical applications. And this is basically taking technology from CERN and trying to bring it towards healthcare delivery. Okay. This slide now summarizes the work or the publications that have been done in my lab for the past 7 years, and I decompose. I'm not going to go through this obviously, but I decompose them into 3 categories. I've investigated a lot of about the impact of dual energy CT for improving radiotherapy treatment planning, and especially with proton planning, because the quantifying the range of the beam is extremely important for this technology. But at the same time, we evaluated the possibility to have more than 2 energies. So we had a dual source CT that was available at the radiotherapy department, but beyond that technology, we had to invent or simulate the information that in order to evaluate the benefits. About 1-2 years ago, I created Lambda Lab, which basically is focus on reconstruction techniques. Okay. So I'm going to summarize three what I find interesting outcome of the research we did. One of them is the so-called eigen decomposition applied to, eigen tissue decomposition. And so eigen decomposition is a quite old technique that is used in linear algebra, and it's also used in quantum physics to describe the states of a system. So you would decompose the state in the sum of eigenstates. So we use a similar approach here, and let me explain it without mathematics. So you see a very nice tiger on the left. The tiger has three channels. The tiger image, pardon me, has three channels, or red, green, and blue. And I'm representing this cluster of points in the red, green, and blue axis that you see. And the idea of eigen decomposition is to rotate the red, green, and blue frame of reference in the direction which describes how the information varies into the image. So you get E1, E2, and E3. And we actually choose E1 to go in the direction, the main direction of how the point varies, and the second direction to be such that the rest of the points are orthogonal, so statistically independent. So the results is what you see here. So eigencolor 1, 2, and 3. So the first one is, mostly the intensity of the image. Eigencolor 2 goes from some purple to orange, and eigencolor 3 goes from green to pink. And you can reconstruct these images either using the 1, 2, or 3 eigencolors. Obviously, if you use 3 eigencolors, you'll get exactly the original image. And if you use 1 eigencolor, you get only one image in some sort of sepia tint. So, but with 2 eigencolors, what's interesting here is you get most of the image. So you choose the first 2 eigencolors and you ignore the third one, you assume it's constant. So if you see the difference between the one with 2 eigencolors and the original one, the only difference is that the tongue is not completely pink and the background is not really green. And in some cases, that can be good enough, so you can reconstruct with less than the full information. You select a fewer number of information, and you can reduce the dimensionality of the problem. And this is exactly what we do with human tissues. So human tissues have the most important elements in human tissue are hydrogen, carbon, nitrogen, oxygen, phosphorus, and calcium. So that gives you 6 degrees of freedom if you include the mass density, and we can use only 2 energies and benefit from the correlations between the elements to get a full portrait of the distribution. And here I just want to mention that this image was taken with a dual source CT some-atom flash, and I'm not pulling any trick on you. This is a purely quantitative pixel by pixel algorithm, so I'm not telling the algorithm that it needs to trace the bone structure. This is really what came out in terms of a quantitative approach. The second approach uses so-called Bayesian statistics. So for the ones who are familiar with Bayes statistics, or for the one who are aware of statistics, you are probably also aware of Bayesian statistics. The idea is to use regularization technique where you would suggest a prior information to your solution. So you would suggest a solution to your system that would be probable. We call that a prior. And I'm showing you an example here on the top is a eigen decomposition technique used on a dual energy CT scan, and the bottom is the one that used Bayesian technique, Bayesian regularization. And you can see that the one in the bottom completely gets rid of the noise, or most of the noise, and we can all see the structures. And by the way, if you're not sure what these are meat boxes. So we did the so-called meat box experiment, animal samples used to measure the range of the proton beam in Boston. and predict them with dual energy CT. And on the right, you can see that we also explore the benefits of multi-energy compared to dual energy CT, and only through Bayesian technique were we able to get rid of the noise and really get the benefits. Last tool I want to discuss is an iterative reconstruction technique. So we combine, at the sonogram level, we use the eigen decomposition model. We also use the Bayesian scheme. And we use an algebraic representation of the measurement system, and we minimize a cost function using the iterative scheme. And you can see on the bottom left the direct filter back projection, so what radon theory prescribes. And on the right, you can see the results of our algorithm using different sets of parameter. And clearly, we can see that we get much more accurate results this way. Okay, so I have just 3 more slides. In July 2018, was published this very nice article about the first ever colored X-ray of a human. So this is a human foot, and it was rendered with colors that represent our daily experience, or at least the ones probably from a surgeon. And I thought it was a very interesting article and also promising technology. I had followed Mars bioimaging since about 2011, and I thought it was quite amazing that they could reach this. So I did some things, and 3 years later, I'm in a lab opening a box. And you probably guess what's inside that box, but let me show you a bit more in detail. So what we did is I sent a student to New Zealand about 6 months later to do a head-to-head comparison between the dual source at the radiotherapy department and the Mars photon counting CT scanner. We used materials which we knew the elemental composition and mass density, and we also used different solutions of iodine and gadolinium. We successfully demonstrated that there were clear advantage of the Mars technology compared to the dual source. And from these results, not only did we get a paper out of this and a collaboration, but we also got a grant for spectral CT mass spectrometry. So we are becoming the first Canadian reference site for the Mars collaboration, and currently the scanner is in commissioning. So I apologize if I'm not going to show you really super interesting results of scans that we have done. It's still in progress. So that is the scanner that was inside the box. So this is the laboratory that in which we installed the scanner. And in the meantime, we are developing an algorithm platform that will have various phases over time. So the idea is to develop our own platform to do analysis of the Mars scanner, but also the raw data. So this is a very special privilege that we get to access to the raw data information. And the rest of the phase will be there to have healthy collaborations and partnerships with the industry or with Mars and other groups. So thank you very much. Here is my group. So there is a lot of diversity in my groups, just that we don't have that spectrum yet in the spectral CT lab. Thank you.
Merci beaucoup, Hugo, pour cette présentation très intéressante. Alors, on va regarder si on a des questions maintenant pour Hugo. Donc, peut-être je peux commencer avec la première question. I'll go in English just for the benefit of Dr. Butler, who should be with us also now. What would be the benefit of having more than 2 energies for tissue characterization?
That's a very good question. So we can demonstrate that if we do a dimensionality analysis of CT data, we get a number between 4 and 5, meaning that we can extract 4 to 5 independent information contained in CT data. And what I mean by that is, as I discussed before, there are 6 elements that are really remarkable in human tissue. But it could go up to 13. The problem is that with the presence of noise, all the differences that you'll see will be sub-Hounsfield unit. So meaning that all the differences will be very much lower than 1 Hounsfield unit. So the idea is to get a more accurate representation of a more accurate characterization of these dimensions. So the idea is to go beyond 2 to reach that potential, full potential.
Okay, thank you. Est-ce qu'on a des questions? Est-ce qu'on m'entend? Oui, ça va maintenant? Donc, est-ce qu'il y a d'autres questions ici? Est-ce qu'on a des questions? Sinon, je peux peut-être en poser une autre. On a encore un petit peu de temps. Donc, is there a theoretical limit as to the number of dimensions that you can extract from CT data?
Yeah, so as I was saying, there's a theoretical limit of 4 to 5, so that depends on the energy. So with soft tissue, you can definitely observe 4 eigen tissue that would describe fully your soft tissue, and while for bones, it's 2. So, so you could say that with 4 energies, it is very likely that you get the full representation of the elemental composition.
Okay, merci. Question de... Est-ce que quelqu'un a des questions aussi dans la salle, peut-être?
I think everybody needs a coffee. Maybe that's the issue at this hour.
Donc, peut-être une autre question. Je pense qu'on a encore quelques minutes. What precision do you expect Mars to be able to reach as a mass spectrometer?
I showed it briefly on the slides. I think we can probably, I don't know if you see this representation well, but the histogram that you see, so I don't have a pointer, but the histograms that you see where you see the elemental composition promises somewhere about 0.5%. So half of a percent, meaning that if, for example, you have a hydrogen content of 10%, you would say 10% plus or minus 0.5. That's my guess, but it's very difficult to answer this question clearly before we have made scans. The other, one of the first work we're going to be doing, and this will be presented by my students, but one of the first we'll be doing is comparison with a mass spectrometer in the chemistry department. So we will be able to really benchmark and determine the accuracy at that point. But at this moment, it's a bit more difficult to determine.
Ah ouais, je vais juste me rapprocher, alors c'est peut-être juste que je suis petite. Merci Hugo pour ta très belle présentation. Tu as montré tout à l'heure une slide avec de la science translationnelle vers la clinique. Est-ce que tu pourrais peut-être commenter brièvement sur le potentiel, puisque dans la thématique là, de cette conférence-là, on parle vraiment de ce parcours-là, le potentiel que cette technologie pourrait avoir éventuellement sur la clinique ou sur les patients?
Merci Ouda pour cette super bonne question. Je ne sais pas si tu veux traduire la question. So I'm going to translate the question. So the question is, can you explain within this translational scheme, what is the potential benefits for radiotherapy? And there are many. Well, I'm very, very focused on dose calculation right now, so really obsessed by the elemental composition because I'm a Monte Carlo specialist. So I like to do Monte Carlo simulations. So we need to determine the electron cross-section, so how the beam interacts with the patient, and we need the element of composition for that. So definitely there will be some improvements in the precision to which you can calculate dose distribution, especially with protons where the beam range is extremely sensitive to these parameters. Now there are probably some other applications that we could get. So for instance, I'm asking you, could you use, if I gave you a map of hydrogen content, could that help you doing your contours? Or could actually that help an AI algorithm to do these contours? Because you get complementary information that you wouldn't see with a conventional CT. So there are some, in my opinion, there are some hidden, or there are some applications we don't even know that this technology has. And this is exactly what our goal is, to actually reach that point and get the best out of it. Also, there's going to be some lateral effects in radiology. It's already undergoing for quantification purposes. For example, the urology or cancer diagnostics and bone damage and so on and so forth. So.
Merci beaucoup, Hugo.
OK. Merci.
Merci encore. Donc, on va continuer avec notre deuxième présentateur ce matin. So it's a great honor to introduce our next speaker this morning, Dr. Anthony Butler. Dr. Anthony Butler is a very impressive bio, so I apologize if I skip some parts because it could be a presentation by itself. So Dr. Butler is a radiologist. He studied physics. He also studied engineering. He's the founder of Mars Bioimaging Limited, a company that does commercialized spectral imaging technology. He's acting as a board member, and he's the president medical of that company. He has more than 150 scientific publications. He holds grants, multi-million-dollar grants, and he's very kind of waking up at night to be with us now and present us about color X-ray for medicine. So without further ado, Dr. Butler.
Well, thank you for having me here, or even if it's only virtually. I'm honoured to be part of this workshop. So today I'm going to be talking about colour X-rays for medicine. So we've just heard from Hugo some very specific applications in radiotherapy. I'm going to give a much more general presentation. I'm doing this on behalf of our team. We've got about 50 people here in Christchurch, New Zealand, and we've got dozens of collaborators around the world. I currently have 3 academic affiliations: University of Otago, where I'm Professor of Radiology and Head of Department of Radiology, University of Canterbury, where I'm in the Physics Department, and CERN, where I'm in the Detective Physics Department. On the right of the screen here is a picture of actually my father's wrist, who's been part of our team, co-founded the company with me. It was one of the early Mars scans we took of people. Today's talk, I'm going to give an overview of our team and our motivation, a very brief discussion of X-rays, computed tomography, and color CT or spectral CT, an overview of some of our scanners. I will then talk about some of the medical applications that ourselves and our partners are looking at, and then a route to take this through to community. Getting technology into the hands of users is an incredibly important thing to do. And as a clinician, I want to make sure the technology actually gets to the point where it can benefit my friends and family. So this is our team in New Zealand. We have about 50 people. They're a mix of scientists, engineers, and medical professionals, including surgeons and pathologists. And we use a bunch of local as I said, color X-rays for medicine, and that's that image that I showed earlier. It was a Mars scan of my father's wrist. Quite deliberately, we left a bit of metal in the scan because that will ruin many MRI and CT images of a wrist. Very proud of that picture. It was seen by 40 million people on Twitter in about 3 days. And then I gave an overview and then jumped to our team here. So this is our team here in New Zealand. We're about 50 scientists, engineers, and other people. And as I say, 3 specialist manufacturers. I'm a clinician by training. I started my university career as going to medical school before I did physics and engineering. And I've always been motivated to actually improve the life of my community. And worldwide, there's about 300 million medical CTs taken every year. And at the moment, most of those are single or sometimes dual energy CTs. So that's about 300 million people every year that aren't getting optimum images of their anatomy or their medical problem. So by changing from standard monochromatic to spectral or color CT, there's a huge number of people that can benefit. CT in general is used for joint applications, bone implants, oncology, both in the radiotherapy domain, but also in the diagnosis and monitoring of cancers, cardiovascular disease such as heart disease and stroke, and of course infectious diseases ranging from pneumonia through to TB. So what do I mean by color X-rays? Well, this is just a simple depiction. In 1895, William Röntgen took a picture of his wife's hand using a photographic plate. It measured the total amount of X-rays that was stopped by the hand, and you get an attenuation image. In about 2006, our team was working with Medipix detectors from CERN, and we were able to take this image of a fetal hand. We used 4 X-ray colors to do that and demonstrated that we could measure the difference in X-ray color between the soft tissues and the bone, or the spectral response between bone and soft tissue. This is actually probably overlapping with Hugo a wee bit, but a standard CT has an X-ray source putting out a range of energies, frequencies, or wavelengths. They're attenuated by the object or a patient, and then you have an energy integrating detector to give you 1 point on a mass attenuation or linear attenuation curve. Our system has a range of colors coming out of a standard white source, attenuated, and then a photon counting detector with the energy discrimination to get multiple points on the curve. So our scanners, we've sold our first commercial scanner in around 2011. We actually did our first spectral image of tissue around 2008. By 2011, we had this commercial prototype that we sold to an organization in North America. We continued to refine the technology, take it from where we had in the scanner a maximum energy of 30 keV up to 120 keV. And this extremity scanner, or we also have a different faceplate to make it a lab scanner. And we're working our way to a head and neck scanner. So we initially started with small animals and excised tissues and are now doing clinical trials. Our extremity scanner has a reasonably small footprint. You can see that it's on wheels, and we've been able to scale the software and electronics. In fact, it scales linearly, so we should be able to go to larger scanners as required. We're likely to have a head and neck scanner in the next year or two, and a potentially a hip scanner soon after that. In my university, we actually have a full bore 80 centimeter scanner that we use for experimental purposes that allows us to do heads, abdomens, and pelvises. So these are some of the applications we've got under investigation. Excuse me. We've been working with a hospital in Switzerland called Lausanne University Hospital, looking at gout and pseudo-gout. This finger was excised due to pain, and we did some plain X-rays. On a dual energy scanner, you can identify where some of the crystals are. You stick it on a Mars scanner, you've got much better accuracy at identifying whether you've got the CPPD or MSU crystals. And of course, you've got a much better spatial resolution. We have a spatial resolution of about 80 to 100 micron. This is some work we've done with the University of Pennsylvania. This mouse has gold nanoparticles in its monocytes. Gold has a very unique spectral characteristic. It has a K attenuation edge of about 80 keV. And in this animal, those monocytes are then tracked into the atheroma. So the aortic arch is passing over the bronchi there, and we can see that gold is accumulated in the atheromas. This is an image of an excised atheroma. So someone who had a mini stroke or a TIA. We took this surgical specimen and imaged it on our scanner. We've actually done about 100 plaques like this. And we can see the fat and the water, which we've colored different labels. And actually we can see the iron from a microhemorrhage. So that's where there's been bleeding into the wall of the plaque. With a number of groups, we've been looking at bone, and one of those groups being Oregon Health Sciences. We've been looking at bone health, looking at cortical thickness and trabecular structure. We also have a calcium map. So that's calcium measured in milligrams per mil. So it's a calcium density. This particular wrist has a metal screw in it. And we've been able to remove, not only identify where the metal is, but we've been able to remove the artifact around that. Some more work done here on cartilage imaging with osteoarthritis. You get changes in their glycosaminoglycans, and using ionic contrast media, you're able to measure where the cartilage is being degraded by, and the glycosaminoglycans have decreased. This is some work out of a Hong Kong university, Chinese University of Hong Kong, looking at arthroplasty failure. This person had pain in their knee following a joint replacement. Pain couldn't be diagnosed. They ended up having the knee replacement taken out. And on our scanner, we're very able to see here, this is a photo, this is a scan of the same object, where the plastic is worn away and the metal is worn away. That was completely invisible on standard CT, and of course in MR you couldn't see anything with the metal. We've worked with a number of groups on cell labeling, and this is a cell pellet, and we've used gold nanoparticles attached to monoclonal antibodies. In this particular well of the pellet of the tray, we've got HER2 positive cell lines, which are common in breast cancer, and we've used a monoclonal antibody called Herceptin, labeled to a gold nanoparticle, and it targets those cells. If we use a different monoclonal called rituximab with our gold complex, we don't see the lighting up. So this has gone orange because we know that the HER2 positive surface antigen is present. This application is with partners from University of Maryland. This is a rat bone. They've taken a nanoparticle that it targets areas of bone microfractures or high bone turnover, and it's lit up, we've been able to light up the bone where they've damaged it. So in this animal, they've opened up the leg, scraped the bone, done damage to the bone, closed the animal up, and then injected this contrast agent into the tail vein, and the agent has targeted an area of microfractures. So that's molecular imaging, something you'd normally do on a bone scan, but done on a CT scanner. So how do we take this sort of technology to the community? This is our first scanner that we've got for point of care. There's been quite a change in medicine over the last 5 to 10 years, and it's really encapsulated in a report out of the NHS in the UK last year, where the UK wants to double the amount of medical imaging going on, but they want that all that increase in imaging that they're going to do over the next 5 to 10 years to be in community clinics, to keep people out of hospital, and to have point of care systems. So our first clinical scanner is a point of care system where you can put an arm into it. Wrist injuries are very common, very expensive, and very difficult for the patient. And normally you have plain films, and then they have to go on and be, if that misses the injury, about 15% do, you have to go on and be imaged with an MRI. So we would retrieve our images on a scanner, do our recombination, have a workstation, and then we place the images in the cloud for radiologist review. So this is the first scanner we placed in a clinic in New Zealand. We've been trialing that since about February this year with real patients. This is a scanner that went to CRCHUM in Switzerland, a hospital there. Some excited radiologists and physicists starting a trial on crystal arthropathies. So in our first, section of the trial, first phase of our New Zealand trials, we did about 19 patients who had fallen on an outstretched hand, looking at the various bones. The sort of images we get, we use 5 energy channels. and we've got a calcium map. And as I say, that's milligrams per mil. So there's our first energy channel. And then we fuse our energy and calcium map together to give an idea of calcium density. We also get a lipid map. So that's a lipid map fused on top of the water map to identify where the fat is in the wrist. And of course, you can get a 3D rendering. If you combine your images correctly, on the left, we have an MRI of a wrist, and here's the Mars image of the same person. And you can see the soft tissue, ligaments and tendons, which, as I say, takes CT to the next level. Instead of CT being a purely anatomical type modality, you're now able to start to determine the materials with fat and water and calcium. Here's another one, I think, just really does show that here we've got a T1 weighted MRI, and on a Mars image, if you look at the fat map, you get basically the same image. So this is a simple case to start one with an occult scaphoid fracture. So this person fell over, hurt their wrist, turned up to the clinic with pain. This is the plain radiograph, nothing could be seen. They went on and had an MRI. In this case, we did a Mars scan at the same time, and just using the very high resolution in 3D nature, you can quite happily see the fracture in there. You can see it on the 3D rendering. So that's simply using the fact that it's a high resolution 3D scan, you can see the fracture. This person had a scaphoid surgery, excision, and then they had a metal pin placed through their capitolunate joint to fuse it. The cone beam CT is on the left-hand side here, and the right-hand side of the Mars images. And I've chosen this because actually it shows how well you can get rid of that metal artifact. You can see the bone metal interface really nicely, which is something the surgeon needs to know. On a standard CT, you get a lucency around it and a lot of streak artifact from it. Again, wrist injury, fallen outstretched hand, triquetral fracture. Standard CT is reasonably low resolution. The Mars image is much, much higher resolution. Case 4 is an avascular lunate fracture. Mars image down the bottom, cone beam on the top. Again, you can see the very high resolution, the ability to see inside the metal screw and the bone metal interface. And using a lipid map, we can actually see that there's been an area of absorption and fat replacement in that particular patient. So in summary, biomedical research has actually demonstrated a very wide range of applications, ranging from cardiovascular through to oncology, some molecular type applications with cell line identification and metabolism, bone metabolism able to be measured, and changes in bone and joint cartilage. So we've got a wide range of applications. We started with clinical trials of wrist imaging, so we can take the scanner into the community where the patients are, rather than having to bring the patients into the hospital. And there's a pathway towards a range of portable scanners to make the clinicians and the patient's life easier. So thank you very much. I'd happily welcome some questions.
Thank you very much, Dr. Butler. Those are stunning images, very fascinating technology. So do we have questions? Maybe I will start with the first question. What do you think is the biggest challenge to make that technology available for the clinic?
That's actually quite a good question. I suspect the biggest challenge is that at the moment, the technology is relatively expensive. So there are lots of groups doing photon counting, and it's likely they're going to bring in very high-end, very expensive scanners. And I think that poses a real challenge in that instead of coming into the medical market with a new technology that's easily accessible, you're going to end up with something like a PET CT scanner, which is really so expensive it can only be used in high-end applications. I think, therefore, the challenge is going to be to put the technology in a setting that is useful for the patients, where the cost can be reduced, where the access to the technology can be much, much better. I like to often compare it to the adoption of PET. PET imaging has been around for ages. It's brilliant technology, but if I look at New Zealand, most rural New Zealanders don't have access to a PET scanner. It's 50 or 100 miles from where they live. They have to have their scan booked in advance. They've got to get the isotope ready. So the access is a huge problem. And I think one of the challenges with spectral CT is going to be reduced price and ease of use. It's not so much an engineering or a physics problem, but more about packaging the technology up in a way that's useful for patients.
Thank you. So we have a comment and question from the audience, from Eli Bouassi. Excellent presentation, thank you. How does Mars compare to traditional CT in terms of amount of radiation?
What we've done is tried to keep the radiation about the same and maximize information. So in many imaging settings, you've got the choice of dialing down the radiation dose, but you increase the noise. So we've focused on keeping the dose about the same and maximizing the information. We actually have a CTDI about half of the New Zealand reference standard, which is fairly similar to the European reference standards. So we're currently using half the dose. We could reduce the dose, X-ray dose further, but you then start losing some of that spectral information. So it's always going to be a trade-off between the two. For the application we've chosen for our first clinical application in the wrist, you've got low radiation sensitive organs. You don't actually have a very big block of tissue to get through. So a CT scan or a Mars scan of a wrist has less than 50 microsieverts of dose. So that's microsieverts. Most X-ray imaging we do in a hospital, we measure millisieverts. So to do a wrist scan like I've got on the screen there is equivalent of a few hours of a long haul plane flight, a few days of background radiation. It goes up, of course, if you start imaging things like the thyroid gland.
Thank you very much. Any other question? I have another question. Are there any upcoming CERN technologies that could also be used for CT? Say a new version of Medipic or anything else you could talk about or mention?
Yeah, I didn't talk about all the technologies under review. So this question was specifically about CERN. So CERN has been working on Medipix 4, which is we're using Medipix 3 in our scanner at the moment. So when my University of Canterbury had, I've been working with CERN on Medipix 4 for about 6 or 7 years now. It has more energy bins. It is able to count at much higher rates or higher photon flux. It has a scheme for correcting the pileup when photons arrive at the same time. So that allows you to go much, much faster again. And it has better energy resolution. It has better correction for interpixel variation, etc. So there's that level of technology change coming out in the next generation of Medipix. There are new tracking technologies coming out, such as TimePix, which allows you to reconstruct energy events much more accurately. It does it instead of having a pulse and then counting how many photons are above a particular height, like a pulse height analysis. It measures the length of a pulse over multiple pixels and allows you to split the energy bins up offline. And I've seen experiments demonstrating that if you can produce 100-point spectrum between, say, 10 kV and 120 kV. So instead of having 10 energy bins, you might have 100 or 1,000 energy bins. And you can start doing more and deep analysis of what happens when 2 photons arrive in the sensor at once, that sort of pile-up region. So that's what's happening on the detector side. There are also changes in the sensor layer. So at the moment, we use a material called cadmium zinc telluride. In fact, most of that comes from Vancouver that we use, on the other side of your country. But we've also used other materials like gallium arsenide and cadtel. And there are perovskite sensors which is a different technology for converting the X-ray into an electrical pulse. And they have some advantages such as clearance of electron hole clouds and the ability to tune where what materials you're using for doping and what effect they have on the spectrum. There's a whole lot of new technologies coming out around reconstruction, and I think these are going to be fascinating. How do we maximize the amount of knowledge we can get out of the data? Hugo talked about some of those as Eigen technique, for example, as a great one. And then built onto the top of that at the end user radiologist end, of course, we've got the AI and the diagnostic support, which is going to alter the way we look through images as well. So I think every part of the CT chain is going to undergo radical improvements over the next 10 or 20 years. And of course, there are things we still don't measure in the X-ray beam, such as phase. So things like phase contrast imaging have been demonstrated quite frequently in physics labs now. Eventually, they'll make it through to the clinic as well. So that's information in the X-ray beam that after it's passed through a patient that we don't routinely record yet. So there's a whole heap of things that are going to improve in my lifetime, which actually I find kind of fascinating because, of course, when you learn about something, you think this is the cutting edge. And then as you get more knowledgeable, you realize that the future is very, very exciting.
Thank you. We still have a bit of time, so I will take the opportunity to ask another question. So you had that slide with the progression of your prototypes. So I'm curious on how long do you foresee the last prototype, human size, being available for clinical trials?
That's a really good question. Again, so we actually have a large prototype in our lab, an 80 centimeter bore that will do a whole human. And we've had that running for a couple of years. In terms of having prototypes like that for head and neck scanning, we could build one of those now. It's essentially a funding issue. What we've decided to do is step through the medical market with a high value, simple system where we can get it out to patients reasonably early. As I said, I think one of the huge challenges for the commercial manufacturers making CTs is with expensive technology, you end up introducing at the top end of the market. And so the medical benefit is there, but the cost benefit just makes it out of reach for most patients, and that slows down the adoption of technology. And coming in at the lower end of the market with a simpler product where doctors can access it more readily actually allows for faster adoption.
Okay, thank you. Any other question? Last chance. Here we go.
Hello, Anthony. This is Hugo. Do you have, thank you for this very interesting talk. Do you have any idea of how much would cost a human-sized spectral CT for radiotherapy, for example? Are we talking about, twice the price of a conventional CT, or 5 times, or 10 times?
Because you end up amortizing your cost, right? So the human one we built in New Zealand, full size, took about 4 years and cost about 12 million New Zealand. What's that, about 8 million Canadian? But that was a one-off. We could replicate that for significantly less, of course. But by the time you then add in all the costs of bringing it to market in terms of FDA clearance, productization, they're going to be in the several million. To have what I think you're after, which is a research one for your lab where it's rather than mass produced, it can be done for a few million. It's achievable. And we've demonstrated the technology can scale to that point.
Thank you. Okay, so thank you again very much, Dr. Butler, for this excellent presentation. And we will continue with students' presentation from Hugo's lab, from Dr. Bouchard's lab. So we'll start with Mohsen. So Mohsen Beikali Soltani, he's a PhD student in Hugo Bouchard's lab.
Okay. Thank you very much. My presentation title and my project title is Tissue Characterization with Spectral Computed Tomography. At first, I will review rapidly traditional decomposition technique and then come back to eigentissue, decomposition and application, and my project. Traditional decomposition technique use interaction-based or material-based. In interaction-based, the original images decomposed into photoelectric and Compton scattering parts. And in material-based approach, original images decomposed into material-specific images. If we have more energy measurement and information, like photon counting CT, we can add other terms, including contrast agents, red terms, in the equation. These methods have advantages in radiology, including virtual monoenergetic images and contrast and non-contrast images. But since these space materials are not the most efficient choice for describing all human tissue, we have limitation in quantification purposes. Also, this method cannot provide the direct estimation of elemental weights, so we have also limitation in estimation of some of the radiotherapy-related parameters. These limitations are resolved with eigentissue decomposition, or ETD, which use virtual material for decomposition. The main idea of the ETD method is that elemental weights in human tissue are strongly correlated. For example, carbon and oxygen are anti-correlated in soft tissue, and calcium and phosphorus are correlated in bone. So, with applying principal component analysis, the dimension of human tissue reduced from 12 in elemental space to K in principal component space, and K is equal 4 for soft tissue and 2 for bone. So we can solve the problem in principal component coordinates and then come back to elemental coordinates to extract elemental weight. This approach was extended to include contrast agents and was modified with Bayesian method to reduce noise effect and tissue fluctuation. With this method, we can obtain map of each element, and also we can estimate radiotherapy-related parameters. In the near future, we will apply ETD method and use the Mars scanner as a mass spectrometer to extract elemental weight of certified reference material of known chemical composition and animal soft tissue and bone sample. Also, for validation, we will use laser ablation ICP device of chemistry department, which can provide an accurate two-dimensional map of elements with high spatial resolution. Apart from application in radiology and estimation of radiation therapy-related parameters, elemental weight can be used as an input for Monte Carlo dose calculation. Moreover, various studies have shown the capability of the Mars scanner for molecular imaging with nanoparticles. Using ETD method, we can quantify several nanoparticles at the same time, and this can be used for evaluation of drug delivery and tumor targeting. Raw data and sinogram data is not accessible for most of the clinical scanners, so the previous ETD method was introduced to be performed at image level, but reconstructed images are affected with beam hardening artifact, and so inaccuracy number can lead to error in estimation of elemental weights. Fortunately, we have access to raw data for the Mars scanner, and we want to reformulate our ETD method to be performed at sinogram level data. And we expect to have improvement in estimation of elemental weight. Thank you. Thank you very much.
Any questions? Non. On va passer à Étienne, prochain présentateur. Thank you. Donc, Étienne Delisle est aussi étudiant en doctorat au labo de Dr. Bouchard.
I'll do my presentation in English, just so that it fits with what Mohsen presented. But to add to the Mohsen's presentation, I'll talk about the Lambda Lab research program, but more on the computational side. So I'll talk about the spectral CT image reconstruction methods. And so the first project I'm going to talk about is essentially the development of the Lambda Lab numerical simulation environment for spectral CT. And this project had as a goal to produce realistic synthetic CT images that modeled all the physics of the CT imaging pipeline and that of course produced spectral CT images. And then the second goal of the project was to do some tissue characterization on those synthetic CT images such that we can test our new methods for tissue characterization. Now you can see the schematic of essentially the whole project, and there are two branches. The first one being a branch that really models all the physics to get the synthetic CT images, and then the second branch does tissue characterization. But what's nice is at the spot where it's green, where we have synthetic CT, we can also use clinical dual energy CT images and then do some tissue characterization on those clinical images. So it's a library that helps us develop new physics, new algorithms, but it's also a library that we can use to do collaborations with the clinic. Now, talking about new methods for tissue characterization, we're working on a pre-reconstruction tissue characterization method that Hugo touched on during his presentation. But essentially what we want to do is take the raw polyenergetic CT data, which is on the bottom left of the slides, and do the tissue characterization on them and then reconstruct those raw tissue characterization maps into the elemental composition maps. And doing it this way, we can really have a better control on the beam hardening artifacts that are always present in our tissue characterization maps, and that really have a large impact on the precision of the quantitative information that we're trying to extract from our spectral CT scans. Now, of course, this is a very difficult problem, which is why it hasn't been done before. Mainly, it's because it's an ill-defined problem that's hard to regularize. So essentially, there are multiple solutions to the problem, and it's difficult to find the solution that matches what we should actually get in real life. So to do that, we use a quantitative prior and a spatial prior that both ask two questions and try to inject our prior knowledge of the problem into the algorithm such that we can constrain the elemental composition maps and get some really nice precision. Now, of course, to do this algorithm works and has been developed in a simulation setting that's kind of precise, but now we have to test it on real data. So we have to do the experimental validation of the algorithm. And as Hugo said, we're very lucky to have a Mars Microlab scanner in our lab. And the goal will be to apply the pre-reconstruction tissue characterization method on the Mars Microlab scanner. Now, of course, to do that, we're going to have to learn a bit more about the potential problems that are going to crop up when we try to apply the algorithm. And so we'll need to do some Monte Carlo modeling of the scanner, and then we'll be able to apply CIRAS directly on the data. Now, for the Monte Carlo modeling, we have a summer student that has been working on that a lot and has done a lot of work, and the simulation helped us understand the scanner very well, and we'll be able to include that in our application. And finally, when the time comes to apply CIRAS on the scanner data, then we have multiple challenges that we think we'll have to deal with, mainly challenges that are that focus on ensuring we have precise results. So we have very nice, a nice precision on the elemental composition maps because that's essentially the end goal. And to do that, we'll have to understand the errors that come from all the different manipulations that we have to apply during the production of the tissue characterization maps. So that brings an end to my presentation. So I can take questions in English or French.
Merci beaucoup. Est-ce qu'on peut aller aux questions? Est-ce qu'il y a des questions pour Étienne et Mohsen? Non, peut-être une question pour Étienne. So how much do you think the Monte Carlo model will be helpful in the implementation of the CIRRUS algorithm?
Yeah, so the goal of the Monte Carlo model is really to have an idea of the uncertainty that we'll get when we do the imaging process. So we're trying to model the imaging process. And I think it'll be useful to understand the sensitivity of the algorithm, the reconstruction algorithm, to the more realistic images, because the algorithm was tested in a simulation setting, which is all perfect and everything. But when we have a Monte Carlo model, then we have to take into account the geometry and how the geometry affects the reconstruction. So I think it'll be helpful to understand what kind of problems we'll get when we move to the real clinical implementation.
Okay, thank you. C'est bon? C'est bon, alors on va en profiter de peut-être remercier une dernière fois nos présentateurs pour les excellentes présentations de cette première session.
Bonjour à tous. Eh bien, voilà, on m'entend bien. Mais alors, je suis Sabri Rial, stagiaire postdoctoral au CRCHUM dans l'axe cardio-métabolique. Et je suis très honoré aujourd'hui d'introduire cette nouvelle session des 12e journées scientifiques consacrée aux neurosciences pour mettre un peu de lumière sur ce domaine fascinant de la neuroscience. Pour commencer, nous avons le grand plaisir de recevoir Ciaran Murphy-Royal. Docteur Murphy-Royal, bien merci d'être là. Il est professeur adjoint à l'Université de Montréal, département neurosciences, et chercheur également régulier au centre de recherche du CHUM. L'équipe de Dr Ciaran Murphy-Royal s'intéresse aux populations neurales, aux interactions entre ces cellules-là et aussi à comment le stress impacte leur physiologie et leur communication. Et en bout de ligne, quel impact ça a sur le comportement en général. So, Dr Ciaran Murphy-Royal, please, c'est à vous.
Merci, Sabri. So I'm going to present today in English my work from my lab on astrocytes, so a type of brain cell. And so astrocytes, the kind of name reminds you of a star, so the title is Twinkle, Little Star. And I'm going to tell you about some of the new imaging strategies we've been using to study these cells and see what they're actually doing. And so to start, of course, I'd like to acknowledge the team. This is my team here at the CRCHUM. And so, yeah, we've got a good few people now in the lab. We're doing some nice work on astrocytes and looking at all different aspects from the cellular to behavioral scales. And later on, you'll see some work from Aoife, who's going to show you exactly what we've been doing in the lab. And so I'm a cellular neuroscientist, so it makes a big change from the talks from earlier on today. And so we're looking at different types of brain cells and how they communicate and interact with one another. And of course, when you think of the brain, you think of neurons, and you can see a nice big red neuron here. They have a cell body, they got axons, they got projections to different neurons, they form synapses and carry out lots of cellular communication. But there's not only neurons in the brain, there's lots of other cells, of course. We study the astrocytes, these star-looking cells. These interact with other brain cells to carry out, I guess, they contribute to cognition, learning and memory, different sorts of behavioral processes too. But you also have microglial cells, the resident immune cells of the brain, and oligodendrocytes, which form the myelin and the white matter for those of you interested in diseases like multiple sclerosis. And so all of these cells, they interact with one another to carry out all the different processes in the brain. But I'm going to focus today on the astrocytes, so the yellow cells there, and how they interact with the neurons. And so this brings me to the big question that's, I guess, plagued the field for many years. What do astrocytes do, and why should we care? And so this kind of dialogue started back in the early days with Camilo Golgi, who's a neuroanatomist. He would get pieces of human brain tissue, and he would do his Golgi staining, and you would sometimes see these star-like cells here. And so you can see just different types of astrocytes. There's many different ones in the brain. And on the bottom right of that image there, you can see astrocytes that are touching blood vessels. So he had some ideas just from the neuroanatomy, just from the physical structure of these cells, that they might play roles in metabolic support of the brain. They might take away waste. They might be involved in sleep. These are all just guesswork that he had from these pictures that he was making. Later on, we had Santiago Ramon y Cajal, who was another neuroanatomist who had an even nicer staining approach, and he took pieces of human brain and also found astrocytes in there, kind of drawing almost the same pictures that Golgi would see. And Ramon y Cajal at the beginning thought that astrocytes weren't really doing much and maybe just forming some structural support, kind of holding the brain together. And later in his career, he kind of came around to the idea that these cells might actually be doing something. And so to kind of bring this kind of a Montreal flavor to this talk, lots of people are interested in astrocytes, including Dr. Penfield here, who we know that started the Montreal Neurological Institute. So he kind of pretty much founded modern neuroscience research as we know it in Canada. And Wilder Penfield, while he didn't really work on astrocytes too much, he did have a nice boat that he named the Astrocyte 2. So this is a picture of him sailing his boat in the Eastern Townships here. And I don't know what happened to the Astrocyte 1 boat, but this was number 2. So astrocytes in the brain, so what do they look like? So those early images were taken just from some guys looking down microscopes and drawing pictures, but now we can actually use different microscopy techniques to see these cells in the brain. And so if you take this brainbow mouse, so in this case they just randomly express different colors in astrocytes, you can see just in this picture there's lots of different colors of astrocytes, and they completely line the whole brain. So they're everywhere. There's not a place in the brain that doesn't have an astrocyte in it. And there's as many astrocytes in the brain as you would have neurons. So there are quite a few of these cells. When we take a look at the single cell level, so we're looking at individual astrocytes here, some in red and some in green. And what you'll see is that they have this cell body, they have this bushy, like big processes and then bushy processes. So they're more like a sponge rather than a star. And so when you look down at the level of the synapse, in red here you can see a dendritic spine with synapses kind of coming off the sides. In green you see an astrocyte, and astrocytes are really getting right into the synaptic element and participating in all of these synaptic communication. You think about plasticity, you think about learning and memory. Everything that goes on at the synapses, the astrocytes are listening to and contributing to. And so on the electron microscopy scale, to get to the finest scale that we can see right now, you can see the presynaptic element releasing neurotransmitters in yellow, a postsynaptic element, so receiving the signals, that's the second neuron. And in pink, you can see the astrocyte that's getting right into this chemical synapse. So they're there, but what do they actually do? Well, some of the ideas that were brought up earlier is that astrocytes might be participating in cerebral metabolism. So if you think about the brain, this is a kind of an image of a human brain, and looking at all the blood vessels, well, there's a highly vascularized organ and uses up a lot of energy for its size. When we look at astrocytes, if you just label them with fluorescent dyes, what you see is that while we're just labeling astrocytes here, you can see blood vessels. So these cells are reaching out processes which completely wrap the cerebrovasculature, and in this way they actually can control blood flow in the brain. And this is important because when you think about energy getting to the synapses, so here you can see a schematic diagram on the bottom left. There's a blood vessel, there's some blue astrocytes touching the blood vessel, but there's some synapses you can see in red that are far away from the blood. So how does the glucose and all the energy substrates get from the blood to these synapses? Well, it goes through the astrocytes. So they're all connected together in these networks that allow them to shuttle metabolites such as glucose to the synapses. And then actually during sleep, astrocytes are really important in clearing away waste from the brain. So it's another kind of important process. And so then it gets us, okay, we got all these kind of, I guess, structural and physical properties of astrocytes that informs potentially on their function, but it's really hard to understand what astrocytes do without us seeing them do it in real time. And so one of the, I guess, pivotal technologies that's really changed the field of astrocyte and neuroglia interactions was the advent of genetically encoded calcium indicators. So astrocytes don't have action potentials, they don't do any electrical signaling as we know it, kind of from neurons, but they do have lots of calcium as transients. And so GCAMP was invented from GFP. So this is on the left, you can see a GFP molecule, which was isolated from the jellyfish, and also won a Nobel Prize in 2008 in chemistry. You can see Roger Chen here, who was one of the founding, I guess, people who discovered and really brought GFP and GCAMP to the field. And so from this GCAMP or GFP molecule, we now have this GCAMP, which is kind of a mutated form of the green fluorescent protein. And so this protein can bind calcium, and when it does, it changes fluorescence. And so every time we get some calcium activities in astrocytes, we can actually see it happen in real time. And this is a really great technical tool to have in the field because it's genetic. We can target it specifically to the cells that we want. So we can use promoters or transgenic mouse lines, so it's just in a cell of interest or a place within the cell. So the expression is non-invasive. We don't have to open up the brain and pierce the cell and put a dye into the cell. You can do it in intact brain tissue in mice and rats, but it has slow implementation. So you need this genetic protein to be overexpressed and mature, and it's quite big. So if you express too much of it, it can be a little bit toxic. So you got to be somewhat careful when using GCAMP. And so we have this nice fluorescent dye that we can use now to see astrocyte function, but we still need a nice way to see it. And so the next big technological development was 2-photon laser scanning microscopy. So for those of you who know confocal microscopy, it's a similar principle. Apart from on the left here, you can see single photon excitation. You shine a laser or a light source, let's say at 400 nanometers, and you get the green fluorescence at 500 nanometers. With 2-photon microscopy, you actually use a higher wavelength, so that's lower power, so it's not burning the tissue as much. And you pulse the laser really fast, and 2 photons hit the same point at the same time, then you will get fluorescence of your molecule. So it's just, it's a similar, in principle it's similar, but there's a lot of advantages to 2-photon microscopy compared to confocal. One of the principal ones is the precision of the excitation spot. So here you can see, it's a pity I don't have a laser, but here you can see in the middle there's a green dye, and there's 2 objectives, one on the left and one on the right. The objective on the left, you can see it's shining light into the green sample. You can see it forms kind of a long cone, so you get excitation above and below the focal plane, so you're focusing on cells in a single plane. With confocal microscopy, you're exciting actually a lot of the tissue. And on the right-hand side, you can see a different objective. In the middle, there's a single point, a little dot, and that's what it looks like with 2-photon excitation. So you're able to hit a single point in your tissue sample, could be brain, could be any piece of tissue, or even a dye in this case, and you're only exciting the molecules in the exact focal plane. So it's really great technology. And so with this, it's kind of really changed how we look at the brain and what we do. Here's an example this is in a mouse, fixed brain tissue. They just have a neuron stained with GFP. And you can see that using this 2-photon laser scanning microscopy, we can go actually really deep into the brain. So going down to about 1 millimeter, 1.5 millimeters, and getting still really nice cellular resolution was unheard of before 2-photon microscopy. And so now we can use this kind of imaging strategies, confocal, as well as 2-photon, to look at astrocyte activity, so calcium activity using GCAMP in astrocytes. So in 2010 was one of the first examples that I could find looking at astrocyte calcium activity with membrane-targeted GCAMP molecules. This was from Baljit Kak's lab in UCLA. And he had cultured astrocytes, and he saw that when he gave synaptic stimulations, so he saw that astrocyte calcium would change. So these cells seem to be listening to synaptic activity and responding with calcium transients. And so with 1 action potential, so very moderate stimulation, you got a little bit of calcium activity in astrocytes, and with 90 action potentials, you got a lot of calcium activity. And so interestingly, these cells in culture, they didn't respond as like a whole, like a kind of like a blob. Actually, they responded with these microdomains. So you can see there's individual places in the cell which are independent from one another and have independent calcium activities. So this kind of suggests that maybe there's, kind of compartmentalization of astrocyte function, listening to individual synapses independently. And so we take this technology to today, or the present modern day, I guess, and we can see that we've come a long way now looking at astrocyte calcium dynamics at the single cell level. So here we can see this is brain tissue, not in the brain itself, but you can see with this movie, there we go, on the left, you can see really nice kind of astrocyte morphology and these great calcium dynamics that occur spontaneously as they're listening to synaptic activity. On the right panel, you can see all these colors, and that's actually, if we could replay that again, that's actually a machine learning algorithm which helps you analyze and pick up the calcium activity in astrocytes. So when I started doing these kinds of imaging approaches and analysis, for me, if I did maybe half a day of imaging, I'd probably spend a week or a week and a half actually analyzing the calcium data almost by eye or by hand, looking at the calcium traces. And now we have machine learning algorithms that do all the work for us, so it's really great. And so in my lab, we've been using recent advances in GCAM technology, so GCAM 6, so the 6th version, F being fast, in astrocytes in the amygdala. So we're really interested in stress, how astrocytes are involved in stress detection and stress disorders leading to, let's say, anxiety and depression. And so we've been looking in the amygdala and these kinds of brain regions. And you can see work from Anthony in my lab. He's looked at GCAM and astrocytes in the amygdala and used the same machine learning algorithms to really see how active these cells are and what they're doing. And so we know that so we got this nice calcium activity in astrocytes, but what's it really doing? What's it important for? So astrocytes are actually able to influence and actually control synaptic activity. So as I showed you before, astrocytes are right into the synaptic cleft. They get in where the business end of the neurons are. And so we think about these astrocyte calcium activities. They seem kind of random and all over the place, but actually with even newer technologies for super resolution microscopy, we're able to look at astrocyte calcium activity at the single synapse. So in pink here, you can see a dendrite. On the sides, these kind of little balls are synapses themselves. In green, it's an astrocyte, and you can see the heat map of the astrocyte calcium over time, showing that these astrocyte calcium activities are actually occurring at the synapses, so they're right where they need to be. And we've known actually for quite some time before this that astrocyte calcium is important. If you look at synaptic plasticity, so this is a process believed to underlie learning and memory in the brain, so where you get synaptic activity strengthening the communication between synapses. In green, these green dots, you can see you have basically synaptic activity as a baseline. Where the red arrow on the bottom is, you're giving synaptic stimulation and you get this strengthening. So the synaptic transmission is increased, you get strengthening of synapses, and if we block astrocyte calcium activity, we no longer get this strengthening of synapses. So this has been shown to be extremely important for processes such as learning and memory. So astrocyte calcium is not just something that looks great and is nice to image, it's actually important for something. And so getting back to one of the, I guess, fundamental properties of astrocytes and what they do is they regulate brain blood flow. And so I took some examples from a colleague of mine in Calgary. I was actually in this lab for my postdoc, Grant Gordon's lab, and they have this nice paper now in bioarchives where they're looking at astrocyte control of blood flow in vivo, so in awake behaving mice. So they take a mouse, they install a head bar, they give it some training on the microscope, then they'll do a cranial window in the skull, so basically you're drilling down to either thin the skull or make a hole in the skull and put a cover slip on top. And then you can let the mouse recover. You put it on an air-supported ball, so the animal's able to run around and behave normally. It's fully awake. You can give whisker stimulations and look at the responding changes in blood flow in the somatosensory cortex. So if you look at stimulate the contralateral whiskers, on the other side of the brain, you will see neuronal and synaptic activity. And so Adam sent me some really nice images, the first author here, of what this looks like. So that you can see in this image I'm going to show, in red is a blood vessel, and in green you can see astrocytes expressing GCAMP. And so the blood vessels have been labeled as red dye with a tail vein injection, and the GCAMP is expressed transgenically. So in this image here, this is in vivo, so in awake, a live mouse. You can see these really nice big red blood vessels at the top. It's a penetrate, or the arterioles that descend into the brain. In green, you can see all these lovely GCAMP expressing astrocytes. So you get some really cool images nowadays with this kind of technology taken with 2-photon microscopy. And so Adam then wanted to see how astrocytes actually are involved in this blood flow regulation. So he was, in this image here, you can see in that big red circle, that's a blood vessel. In green, it's an astrocyte expressing GCAMP, and he gives a whisker stimulation. And so you can see there's lots of movement here because the animal is awake and running on a ball. And so when you can see when we give this really nice whisker stimulation, the astrocyte lights up, and you can see changes in the blood vessels. Can we play it again? Perfect. So you can see the whisker stim comes on, there's lots of astrocyte calcium, and the blood vessel dilates. And so you can see this here in this kind of summary data at the top is before the stimulation. You can see the bar shows the width of the vessel. As the stimulation goes on at 6 seconds, 35 seconds, the blood vessel is getting much bigger and goes back to normal at the end of the stimulation. And so in this work that Adam has on bioRxiv, they showed that these blood vessel diameter changes, these really long changes in blood vessel diameter, are they're sustained by astrocytes. So I'm not going to go into the data, but if you block astrocyte calcium signaling, you no longer get these sustained long changes in blood flow to the brain. So it's quite an important element in that. And so lastly here, I'm just going to talk a little bit about the work that we've started and that's ongoing in our lab. So we got, of course, the synaptic work. You saw the blood flow and the blood vessel stuff. And now we're trying to take it to another level and look at how astrocytes are involved in behavior. So we're using a slightly different approach here. We're using still photometry. We're looking at fiber photometry here, where we excite GCamp in awake behaving mice. So we have some LEDs. We shine, blue and violet light into the brain. We get the green signal back. And so rather than get cellular resolution here, we only just see changes in fluorescence of the GCamp signal in the brain region of interest. And so although we can't see the single cells, we have the advantage of allowing the mice to be tethered with a small little wire on their head, and they're able to freely move in any behavioral arena. So this is a mouse in the home cage with the fiber attached to its head and a fiber optic implant going into the amygdala, so the brain region that's associated with threat detection. And so some work that I've done is looking at this in different behavioral arenas. So this is really, a preliminary work here where we see a mouse on an elevated plus maze. So when the mouse is in the closed arms, so it's got high walls, the mouse feels secure, it's safe. When it goes into the open arms, there's no walls, it's kind of exposed to the room. So it's just a platform in the middle of a room. And when we look at astrocyte calcium signals here, you can see in green, every time the mouse goes into the open arms, we see the astrocyte calcium increases. So it seems that these cells are, contributing to threat detection in this arena. And so even still, we kind of did some learning and memory tasks, and this one is just a simple fear conditioning. So we put mice in a specific environment. So it's a box with a checkerboard wall. We give it foot shocks, and the mouse then associates this place that's never seen before with danger. Of course, it's very threatening. When I get put in here, I'm going to get shocked. So you put the mouse back. So actually at the bottom, then you can see the astrocyte calcium signal. Every time they get a shock, astrocyte calcium goes up. We take the mouse out, we put it back into the box 3 hours later, and you see the astrocyte calcium is quite active. So it's almost as if it's waiting for something, that the threat detection is quite high. And over time, we take it out, put it back in again, take it out, put it back in. You can see at 24 hours, the astrocyte calcium's gone quite low. As the animal realizes, I'm not getting any more shocks in this environment, I don't need to be afraid. So we're kind of developing different strategies to use this kind of learning and memory tests to see how astrocytes are involved in threat detection, how that's affected by stress, etc. And so this is just some examples of the kind of maybe imaging technologies to see astrocytes, see how they work, what they're doing. And so the big questions, there's lots remaining, so what's next? So this is just a nice image of a brain slice with some stains for astrocyte proteins. And so the primary challenge in the astrocyte field is linking these cellular changes to behavioral, cellular observations to behavioral changes. So how can we link what astrocytes are doing on the cellular scale to what's going on when in the cognitive processing. And so there's still not many tools available to manipulate astrocyte function, so they all lack temporal resolution. So the things that we do to astrocytes are somewhat permanent. We turn them off, it's permanent, or we can turn them on, and it's kind of not very fast. And so we're using now nice transgenic mouse lines to specifically target astrocyte function, and with this, the work you'll see from Aoife in a few minutes, we'll look at the impact of stress on astrocytes and how this results in cognitive impairment. And so thank you all for your attention, and thank you for tuning in online too.
Thank you so much, Ciaran. It was really fascinating, as expected, as I said. Are there some questions, maybe here in the room, but also in the Q&A? Oui. Non, il n'y en a pas en ligne, donc tu peux y aller, Vincent.
That was beautiful, Ciaran. Really, really cool data. I have a couple of questions. Do the astrocytes, so you show that they're all connected, right? So they form a network. Is there a sort of a pacemaker activity between them? Do we know that, or do they act sort of on their own independently?
Yeah, so that's they can function independently, but there's lots of, I guess, when there's high levels of, let's say, brain activity, they need to use the network. And so it's kind of a bit of both. And for the basal, I guess just the simple, I guess, basal levels of brain activity, it seems that individual astrocytes are fine. But once you start to crank the system, like do a learning and memory task or try to do something more complex, you really need the network. And it's really dynamic. It's regulated at different times of day, it seems, and also by stress as well. It really impacts the network.
Okay, second question. Thank you. You focused on calcium as a, obviously a reader or readout of electrical activity. There are other sensors available for other second messengers that are important for neuronal activity in general, including cyclic AMP. Have you looked at that? Is it possible to use these sensors in this setting?
Yeah, I think that would be really, really great to do that, because lots of the signaling in astrocytes is through metabotropic receptors. So, I mean, that's something that we really need to get into, is looking at these second messenger pathways. And I guess calcium just shows that something's happening. We don't know what pathway is being activated. So if we could really get a different sensor to really see what's happening, that would, I mean, increase the amount of information. I think that's a really, really great idea.
Dr. Nguyen?
Yeah, nice talk. So have you looked at the effect of medications inducing behavioral side effects on astrocyte activity using your model?
Yeah, so we haven't, but there has been lots of work showing that lots of drugs that I guess are believed to act specifically on neurons actually have lots of effect on astrocytes. So one thing that we're interested in is stress, and so lots of antidepressants, fluoxetine, all these kinds of SSRIs, these other drugs actually have lots of effects on astrocytes. So that's something that we're interested in looking into to see maybe can we isolate the astrocyte response to this drug maybe some way to see if we can target those cells specifically. But yeah, we haven't started that work yet. We're still on the really basic, okay, what are they doing? How are they doing it? And then later we do want to get into, I guess, the technology development.
We have a question from the Q&A first. Dr. Thierry Alquier from CRCHUM said, great talk, Ciaran, first. So what could be the next useful tech development to image astrocytes in vivo and better understand their functions?
Yeah, I guess what's next, what we want. So I guess we want to miniaturize everything. That would be the goal, have a 2-photon microscope that sits on the mouse's head and it's able to walk around and not be head fixed on a ball. So I guess that's the next big thing, and it is coming. People are developing these technologies, and so I think that's where we can go, and we can get the single cell resolution in awake, naturally behaving animals. Then we're going to see some cool stuff.
That would be great. Please.
Yeah, so I have some simple questions, but what's kind of the size scale of these cells? Because I don't know much about these things. So
yeah, so I didn't put a scale bar up. Yeah, they're a lot smaller than neurons. So they're about, I guess, the whole size. The cell body themselves is 10 microns. So they're probably about, I guess, 50 microns across. They're quite small. Well, they're still quite big, but they're not as big as neurons, right? They're not projecting to different brain regions. They're quite local in their activity.
Okay, cool. And I have another question about the calcium concentration. So do you know how much calcium is actually present in those cells? I don't know if it's a measurable difference in concentration.
Yeah, with the GCAMs you can't measure it, but with other dyes, with like inorganic dyes that you would basically pump into the cell with an electrode, then you can start to measure calcium concentrations. But right now, yeah, that's one of the limitations of GCAMP, is looking at the amount. So we can just see, before and after kind of effects or manipulations. We don't know the precise concentration. It's true.
Okay, thanks.
In the Q&A, we have another question from Aurélie Cléré-Bureau. So nice presentation, thanks. Do you have an idea of astrocytes' role in chronic pain? Do you think that they have another behavior? Thanks.
Yeah, so in chronic pain, I know people working in astrocytes in the spinal cord and seeing these kinds of chronic pain models. We do nerve ligations or damage to limbs, and you see these astrocytes. They look like these inflamed astrocytes, so they get much bigger. Their processes get more dense, and you see all of these big morphological changes. And so, yeah, there's really big effects of pain and other things on astrocytes, especially these chronic inflammatory kind of insults. Astrocytes are involved in some way. Yeah.
I do have one other. I think we have some time. It's a question that will sound very naive to you. It's a kind of egg and chicken question concerning the regulation of the blood flow in the brain by astrocytes. There's very nice pictures we show. My question is, what is, do you think, the first occurring? Do you think the blood flow that is known to be regulated upstream by heart, etc., result in signaling within brain, or inversely, there is something via neurons triggering an astrocyte regulation?
So I guess we're very, I guess, brain focused, so we don't even consider anything below the neck, I guess. But yeah, I think in the beginning, the first dilation is supposed to be from neurons. So the neurons release nitric oxide and other vasoactive molecules that cause dilation, and then the astrocytes come on a bit later. So it's kind of, depending on the amount of activity, the astrocytes will come online or not with just small activity. I didn't show in that paper, but if you just give 5 seconds of whisker stim, the neurons turn on, the astrocytes don't. Okay. So they don't need, and there's a small dilation. And then if you give the longer stimulations, then the neurons turn on, the astrocytes turn on, and you get the whole network being activated. So there is, I guess it depends on the event itself, but yeah, there's kind of a somewhat complex. Blood flow and vasodilation in the brain is really a hard thing to study. Yeah.
Oh, great. So, we do have one there. It's very successful. Can we use GCAMP6F, this last version, astrocyte imaging in zebrafish to record stress in epilepsy mutated line? Currently, we have GCAMP6F line in genetically mutated fish. It's from Dr. Uday Kundap.
Oh yeah, so I actually had to shorten my presentation. I had too many slides, but my last slide was going to be about GCAMP and astrocytes in fish. I think that's some of the most exciting work going on now, is actually from fish, and you can really look at the behavior really nicely. So if I had an extra 10 minutes, I could have shown that data. But yeah, GCAMP, astrocytes in fish. There's fish in worms, flies, astrocytes in worms, flies, and mice, rats, humans. So you can really look at them in any model that you want.
Great. I think if we don't have any more questions, so thank you so much, Dr. Ciaran Murphy-Royal. It was really great to hear from this wonderful research. So. À présent, je vous invite donc à accueillir Dr. Dong Khoa Nguyen. Dr. Nguyen est neurologue au CHUM, professeur titulaire au département de neurosciences du CHUM, et il est également chercheur au CRCHUM. Il a plus de... Est-ce qu'on m'entend? Allô? Oui? Oh, d'accord. Il a plus de 150 publications à son actif et quasiment le double en présentation de conférences. Donc, on est heureux de l'accueillir pour cette nouvelle présentation. Merci beaucoup, Dr. Nguyen.
Merci pour l'introduction. Je vais faire ma présentation en français. Donc, merci au comité organisateur de m'avoir invité. Donc, comme il a été dit, je suis neurologue au CHUM, chercheur régulier dans l'axe neurosciences. Il me fait plaisir d'être ici avec vous pour vous parler de spectroscopie proche infrarouge appliquée à l'épilepsie, d'où le titre de ma présentation, « Faire la lumière sur les crises d'épilepsie ». Et puis, voici ma déclaration de conflit d'intérêt. J'ai aucun conflit d'intérêt à déclarer en lien avec cette présentation. J'aimerais aussi dire que je ne suis pas un ingénieur, même si je vais aborder des notions d'ingénierie. Mon père était ingénieur, peut-être que j'ai développé un intérêt à cause de lui, mais je travaille avec des très bons ingénieurs, dont Frédéric Lesage, qui est professeur à l'école polytechnique. Il y a aussi Kay Peng, là, qui est un chercheur qui est postdoc dans mon labo, et puis Ali Kassab, qui est étudiant au doctorat dans mon labo, qui est en neurosciences, mais qui a dû apprendre beaucoup de notions d'ingénierie. Donc, voici un survol de ma présentation, donc une brève introduction sur l'épilepsie et puis sur la spectroscopie proche infrarouge. Comment on est venu à développer nos propres prototypes d'enregistrement de spectroscopie proche infrarouge combinés avec l'électroencéphalographie et comment on a utilisé ces prototypes-là pour étudier les décharges épileptiques et les crises au sein de l'unité d'épilepsie du CHUM et puis également aux soins intensifs. Alors, pour ceux qui ne connaissent pas c'est quoi l'épilepsie, c'est une condition chronique caractérisée par des crises récidivantes. Et puis, c'est une condition assez fréquente. Ça atteint à peu près 50 à 70 millions à travers le monde. C'est la deuxième condition chronique neurologique la plus fréquente. Et puis, c'est quoi une crise? Une crise, c'est un changement de comportement transitoire qui est dû à une décharge neuronale excessive. En gros, là, c'est comme un orage électrique qui se produit au niveau du cerveau. Et puis, les symptômes que les gens vont avoir sont divers. On divise les crises en deux grandes familles. Vous avez les crises focales, ou qu'on appelait auparavant les crises partielles, et puis vous avez les crises généralisées. Les crises focales, c'est des crises qui sont dues à des décharges qui commencent de manière focale dans le cerveau. Donc, ça peut venir, par exemple, du lobe temporal, du lobe frontal, du lobe pariétal ou occipital, que ce soit du côté droit ou gauche. Et puis, les crises d'emblée généralisées, c'est des crises qui prennent tout le cerveau dès le début. Et puis, les gens vont avoir des symptômes qui varient selon le type de décharge, la localisation de la décharge, où est-ce que la décharge se propage, la durée de la décharge et l'intensité de la décharge. C'est pourquoi les crises vont varier d'une personne à l'autre. Donc, le traitement de base de l'épilepsie, c'est les médicaments qu'on appelle des anticonvulsivants. Maintenant, on appelle plutôt ça des médicaments anti-crise. Et puis, malheureusement, c'est à peu près le tiers des patients qui ne vont pas répondre au traitement qu'on a actuellement. Alors, pour ces gens-là qui ne répondent pas au traitement médical, on va utiliser des méthodes alternatives, comme par exemple la diète cétogène et la neuromodulation, comme par exemple le stimulateur du nerf vague ou la stimulation cérébrale profonde. Mais le traitement le plus efficace pour les gens qui ne répondent pas au traitement médical, c'est la chirurgie d'épilepsie. Il y a plusieurs types de chirurgie. La plus commune étant la cortectomie, qui consiste premièrement à identifier le foyer épileptique, donc d'où originent les crises, et le resséquer pour guérir le patient des crises. Alors pour ça, il y a premièrement la première chose à faire, c'est de trouver d'où viennent les crises. C'est le plus important. Et puis après, une fois qu'on l'a trouvé, il faut déterminer si cette région-là est dans une région qui est assez sécuritaire pour être opérée. Il ne faut pas que cette région-là surplombe une région importante du cerveau, comme par exemple les régions langagières, les régions motrices ou les régions visuelles. Sinon, le patient va avoir un déficit neurologique post-opératoire. Donc, les patients passent à travers une batterie de tests, comme par exemple la résonance magnétique cérébrale, pour essayer d'identifier une lésion épileptogène, donc une lésion structurale qui pourrait être responsable des crises. Ils vont passer aussi des enregistrements EEG, des enregistrements électroencéphalographiques, pour essayer d'identifier la zone d'où viennent les décharges électriques anormales. Ils vont aussi avoir une TEP scan, donc une tomographie par émission de positrons, pour identifier la région épileptogène qui est généralement hypométabolique parce que malade. Ils vont également avoir un spectral, donc une tomographie par émission monophotonique pour identifier les régions où il y a une perfusion augmentée au moment d'une crise. Donc ça aussi, ça peut nous aider à identifier le foyer épileptique. La résonance magnétique fonctionnelle, on utilise surtout ça pour identifier les régions éloquentes, donc les régions qui sont importantes à ne pas toucher, comme par exemple les régions langagières et les régions sensitivo-motrices. Donc, chacune de ces techniques a ses avantages et désavantages en termes de résolution spatiale, par exemple, ou résolution temporelle. On va remarquer toutefois que c'est juste le EEG qui peut être effectué au chevet du patient. Et c'est aussi la seule technique qui peut permettre une surveillance en continu du patient. Donc, on peut installer les électrodes au chevet et enregistrer des heures et des jours, ce qu'on ne peut pas faire avec les autres modalités. C'est aussi l'intérêt pour développer la spectroscopie proche infrarouge qui a le potentiel d'être également un outil qu'on peut installer au chevet et permettre une surveillance en continu du cerveau, mais pas en mesurant l'activité cérébrale, mais plutôt en mesurant l'activité hémodynamique du cerveau. Alors, qu'est-ce que c'est la spectroscopie proche infrarouge? C'est une technique qui permet de suivre les changements hémodynamiques du cerveau basés sur les mêmes principes que l'oxymétrie. Donc, comme vous savez, un oxymètre, c'est un petit capteur qu'on met au bout du doigt quand vous rentrez à l'hôpital pour mesurer la saturation sanguine. Et puis, ici, au lieu de mettre ça au bout du doigt, on le met sur la tête. Et puis, au lieu d'en mettre un, on en met plusieurs pour couvrir toute la tête. Donc ici, la lumière dans l'infrarouge proche va réussir à traverser le crâne et pénétrer environ sur une profondeur de 1 à 3 cm. En général, ça va toucher la surface, la superficie du cortex cérébral. Et puis donc, la quantité de lumière qui va être réfléchie, elle va être captée par ce qu'on a des détecteurs qu'on va mettre sur la tête. Et la quantité de photons qui va être réfléchie, ça va dépendre de la quantité d'hémoglobine qu'il y a au niveau du cortex, de la surface du cortex, parce que l'hémoglobine, c'est un puissant chromophore, c'est ce qui absorbe le plus les photons au niveau de la couche corticale. Donc, si on utilise une longueur d'onde qui est plus sensible à l'hémoglobine oxygénée, alors on va pouvoir mesurer les changements en hémoglobine oxygénée. Et puis, si on utilise une longueur d'onde plus sensible à l'hémoglobine désoxygénée, on va pouvoir suivre les changements en hémoglobine désoxygénée. Et puis, si on additionne les deux, on peut avoir l'hémoglobine totale. Et donc, si on mesure en continu, on peut suivre de manière continue les changements en hémoglobine totale, oxygénée et déoxygénée au fil du temps. Alors, pourquoi suivre l'hémoglobine oxygénée ou désoxygénée, c'est intéressant pour l'épilepsie? Eh bien, c'est à cause du principe du couplage neurovasculaire que Ciaran nous a parlé un petit peu. Donc, quand une région du cerveau est activée, il y a automatiquement une augmentation du flot sanguin dans cette région pour soutenir les besoins en oxygène de la région qui travaille. Donc, on peut voir là en bas à droite qu'il y a une augmentation en rouge de la concentration d'hémoglobine oxygénée suite à une activité neuronale. Et puis, l'apport est en général assez suffisant, voire même en excès de ce qu'on a besoin. C'est pour ça que vous voyez la ligne en bleu. Il y a une diminution de l'hémoglobine désoxygénée parce qu'il y a beaucoup plus d'hémoglobine oxygénée qui est livrée à la région qui travaille. Donc, avec un appareil de spectroscopie proche infrarouge qui va mesurer ces changements en hémoglobine, c'est possible de voir les ondes qui sont activées durant une tâche, comme par exemple quand on parle, quand on bouge la main ou quand on ouvre les yeux, mais aussi pendant les crises épileptiques, parce qu'évidemment, durant une crise, les neurones au sein du foyer épileptique déchargent sans arrêt, de manière excessive, donc ils fonctionnent à plein régime. Donc, ils ont besoin de beaucoup d'hémoglobine. Donc, en collaboration avec des ingénieurs de l'École polytechnique, on a travaillé ensemble à améliorer les appareils de spectroscopie proche infrarouge qui étaient disponibles sur le marché. En fait, les appareils étaient très simples. Il y avait en général 2 optotes positionnés au niveau du front, parce que, bon, il n'y a pas de cheveux au niveau du front, c'est plus facile à accéder. Et donc, on pouvait juste mesurer en 2 endroits au niveau du cortex frontal. Nous, notre intérêt, c'était de couvrir toute la tête. Et aussi, on avait besoin d'enregistrer l'électroencéphalogramme en même temps que la spectroscopie proche infrarouge, ce que les appareils commerciaux ne permettaient pas. L'autre chose aussi, c'est que les appareils commerciaux, c'était principalement pour des études neuropsychologiques qui étudiaient le lobe frontal, et puis on faisait des expériences qui duraient 1 heure ou 2. Nous, on avait besoin d'enregistrer sur de longues périodes. Donc, voici quelques prototypes au fil des années. Vous avez le modèle avec un casque rigide en 2006, après un système portable multicanal en 2011, et puis le système portable de haute densité en 2018. Et puis, ça a culminé donc vers le développement d'un casque qui est confortable, portable, avec des optotes qui couvrent toute la tête. et qui permet aussi l'enregistrement en même temps de l'EEG. Et évidemment, l'EEG, c'est important pour nous là, pour détecter les décharges épileptiformes. Donc, ça nous a permis de faire des enregistrements de longue durée à l'unité d'épilepsie du CHUM. Donc, en effet, des enregistrements de continu de longue durée, c'est nécessaire dans mon domaine parce que si on veut enregistrer les crises, on doit enregistrer de longues heures parce que les patients, évidemment, ne vont pas faire des crises sur demande. Les crises surviennent de manière aléatoire. Donc, on peut voir ici un patient qui porte notre prototype le jour comme la nuit. Et puis ici, l'enregistrement d'une crise là, avec le prototype sur la tête. Donc, un des avantages de cet outil-là, comme je dis, c'est d'enregistrer au chevet. Et l'autre avantage, c'est que c'est un petit peu plus permissif au niveau des artefacts, contrairement, par exemple, à si quelqu'un faisait la même chose dans une machine de résonance magnétique fonctionnelle. OK. Donc, grâce à notre setup, là, on a mesuré les changements hémodynamiques survenant avant, pendant et après les crises, différents types de crises, incluant des crises du lobe frontal, des crises du lobe temporal, des crises du cortex postérieur, le lobe pariétal et occipital. Alors, c'est ce que je vais vous montrer un peu au cours des prochaines diapos. Donc, ici, on voit l'enregistrement d'une crise frontale chez un garçon de 10 ans. Ah non, excusez, j'ai sauté une diapo. Je vais passer à la prochaine diapo, puis revenir sur celle-là. OK, donc ici, on voit l'enregistrement d'une crise frontale chez un garçon de 10 ans. On voit en rouge en bas à droite, juste concentrez-vous sur donc, le côté droit de la diapositive. En haut, on voit l'EEG, on voit la crise qui est enregistrée, puis en bas, on voit les images de spectroscopie proche infrarouge. Et là, on regarde simplement les images au niveau du lobe frontal droit. Puis, si vous regardez en rouge-orange, c'est l'augmentation de l'hémoglobine oxygénée au niveau du lobe frontal au moment d'une crise. Et la localisation de cette activation est concordante avec le foyer épileptique qui est identifié avec d'autres techniques, comme par exemple les images à gauche ou si vous regardez les images de la MEG, on identifie également le foyer. On localise également le foyer au niveau du lobe frontal droit. Pendant que les gens, nos patients, portent le casque, on en profite également pour faire des tâches de langage. Donc, ici, tant qu'à attendre la survenue d'une crise, bien, on en profite qui porte le casque, puis on fait des tests neuropsychologiques. Un des tests qu'on fait, par exemple, c'est on essaie d'identifier les ondes du langage, des ondes qu'on doit éviter lors de la chirurgie. Donc, c'est très important de savoir si le langage est latérisé à droite ou à gauche et dans quelle région. Donc, en leur faisant parler, eh bien, évidemment, les régions du cerveau qui sont impliquées dans le langage vont s'activer et puis il y a une augmentation d'hémoglobine oxygénée qu'on peut mesurer ici. Et puis, on est capable de localiser et latériser les ondes du langage. OK. Donc ici, sur une série de patients qui avaient un foyer frontal, donc on a enregistré plusieurs cas, on a pu bel et bien démontrer que la spectroscopie proche infrarouge pouvait localiser adéquatement le foyer épileptique en identifiant la zone d'augmentation maximale de l'hémoglobine oxygénée. Fait qu'ici, on peut voir un autre exemple. Si vous voyez sur la droite ici, la courbe qui est en haut, vous voyez le début d'une crise qui est associée à une augmentation de l'hémoglobine oxygénée qui perdure durant toute la durée de la crise et puis qui va revenir à l'état basal à la fin de la crise. Et puis sur les images en bas de la reconstruction 3D, vous voyez que l'activation maximale en rouge pour l'augmentation de l'hémoglobine oxygénée, elle est principalement au niveau du lobe frontal, débordant au niveau du cortex insulaire. Donc, on dit qu'une image vaut 1000 mots, mais une vidéo, c'est encore mieux. Alors ici, on voit ici l'enregistrement d'une crise de manière dynamique du début à la fin. Ça, c'est la position de certains optodes. Ce n'est pas tous les optodes qui sont installés. Et on enregistre ici une crise frontale. Et puis, vous allez voir ici l'augmentation de l'hémoglobine oxygénée au niveau du cortex frontal droit au moment de la crise et le retour à l'état basal quand la crise se termine. Parce qu'on est capable de couvrir toute la tête, donc de l'avant à l'arrière, on a aussi démontré qu'on pouvait aussi bien détecter les crises qui venaient du cortex postérieur. Donc, on voit ici sur l'image en bas à droite comme les changements au niveau de l'hémoglobine oxygénée et désoxygénée qui sont principalement localisés au niveau des régions postérieures. Et puis aussi, on a démontré que notre prototype pouvait voir les changements hémodynamiques lors de crises temporales. Donc, si vous voyez en bas à gauche l'EEG, c'est difficile à voir, mais vous allez voir qu'il y a une crise électrique au niveau du lobe temporal gauche. Puis en haut, des images du comportement de l'hémoglobine oxygénée au niveau des capteurs qui sont situés au niveau du lobe temporal. Vous avez une augmentation de l'hémoglobine oxygénée et c'est représenté également à droite où on voit l'activation maximale au niveau du lobe temporal droit, puis par la suite la propagation de la décharge du côté gauche. Et puis, ce qui est intéressant dans les crises du lobe temporal qu'on a enregistrées, c'est que si vous regardez du côté gauche et en haut, au niveau des courbes, la courbe en noir, vous allez voir que l'hémoglobine réduite, elle bouge très peu au début, puis par la suite, elle augmente. Alors, je vous ai dit que d'habitude, dans une réponse neurovasculaire normale, l'apport en hémoglobine oxygénée est en excès de la demande pour répondre aux besoins métaboliques. Ici, l'apport en hémoglobine oxygénée semble être insuffisante pour répondre aux besoins métaboliques d'une crise. Et puis, donc, il y a une accumulation d'hémoglobine réduite, ce qui peut être interprété comme lors d'une crise, c'est un phénomène qui est pathologique. Et puis, la réponse hémodynamique est insuffisante, de sorte que le cerveau est en état d'hypoxie relative, ce qui pourrait peut-être expliquer pourquoi nos patients, après une crise, sont confus ou peuvent avoir des déficits neurologiques transitoires qui prennent un certain temps avant de récupérer. Fait que ça, c'est une observation intéressante qu'on va regarder par la suite aux soins intensifs. Et puis, on a regardé aussi si les pointes épileptiformes pouvaient donner des changements hémodynamiques visibles par la spectroscopie proche infrarouge. Et puis, effectivement, même si pour ceux qui ne savent pas c'est quoi des pointes, c'est des décharges électriques excessives, mais qui durent juste quelques millisecondes. Ce n'est pas assez fort pour donner des symptômes, mais nous, on peut les voir sur l'EEG. Et en moyennant ces pointes-là, on est capable de voir effectivement une réponse hémodynamique qui est concordante avec le foyer épileptique du patient. Alors, fort de notre expérience à l'unité d'épilepsie, on a essayé de transposer ce qu'on savait en faisant des enregistrements aux soins intensifs. Et puis, les résultats, là, on a des résultats préliminaires qui ont été acceptés dans Neurimus. C'est ce que je vais vous montrer rapidement ici. Donc, voici notre setup aux soins intensifs. Ça, c'est 2 patients qui ont eu des enregistrements. Vous avez ils portent notre casque avec les fibres, nos optomes, nos fibres optiques, qui permettent l'enregistrement de la spectrométrie proche infrarouge, puis vous avez les électrodes EEG. Et puis, vous voyez ici un exemple, un patient qui a eu une crise électrique. Donc, ce qui est particulier avec les patients aux soins intensifs, c'est que leur cerveau est comme un peu épuisé ou malade quand ils sont atteints de manière aiguë par une maladie critique. Et donc, les crises qu'ils font ont rarement des manifestations cliniques. C'est surtout des crises qu'on peut voir au niveau électrique avec l'électroencéphalogramme. Donc, ils font toutes sortes de décharges, des crises électriques répétées, des pointes épileptiformes répétitives, etc. Donc, si on regarde ici dans les données préliminaires, on a pu démontrer que chez ce patient-là, par exemple, qui faisait plusieurs crises, la durée des crises avait une importance. Avec les crises brèves, on voit une augmentation de l'hémoglobine oxygénée. L'hémoglobine réduite, elle reste stable. Donc, la réponse hémodynamique semble être adéquate. Mais pour des crises plus longues, comme par exemple ici à 20 à 25 secondes, on voit que la réponse hémodynamique est insuffisante, de sorte qu'il y a une augmentation de l'hémoglobine réduite. Et puis, dans le futur, ce qu'on aimerait faire, évidemment, c'est automatiser ça. c'est de développer une interface facile d'utilisation pour l'équipe traitante pour aviser la survenue d'une crise, de son point de départ, de ses régions de propagation, de l'impact de la crise sur l'oxygénation du cerveau, afin que l'équipe puisse intervenir rapidement. Je ne sais pas si vous pouvez jouer la vidéo. Donc, dans cette vidéo-là, on peut voir de quoi ça pourrait avoir l'air sur un écran de monitoring aux soins intensifs. On voit la région en crise qui s'allume en rouge, et puis il y a un clignotant rouge qui indique quand l'oxygénation tissulaire devient critique en dessous d'un certain seuil qu'on déterminerait. Et puis, on est également en train de travailler sur des méthodes pour automatiser la détection de crises épileptiques avec des méthodes d'intelligence artificielle. Alors, on a, dans un premier temps, là, démontré que les données de spectroscopie proche infrarouge pouvaient améliorer la détection des crises mieux que ce qu'on ferait juste avec l'EEG. Alors, ici, là, on montre là, qu'un réseau de neurones récurrents à mémoire court terme, long terme était en mesure de classifier les états de crise des états non-crise. Et puis, les performances étaient meilleures si on utilisait autant les données de spectroscopie proche infrarouge et de l'EEG que juste une des deux techniques. Donc, notre plan au cours des prochaines années, c'est de faciliter et simplifier encore plus l'acquisition, l'analyse des données de spectroscopie proche infrarouge combinée à l'EEG auprès des patients qui sont à risque de faire des crises. Et puis, on veut aussi étudier d'autres applications potentielles de la technique, comme par exemple le suivi de patients avec hémorragie cérébrale sous-organoïdienne qui sont à risque de vasospasme ou d'accidents cérébrovasculaires. Et puis, éventuellement, là, intégrer la technique en milieu clinique. Donc, en conclusion, donc, la spectroscopie proche infrarouge fonctionnelle est capable d'effectuer une surveillance non invasive à long terme et au chevet du patient. Elle a, j'espère que j'ai démontré qu'elle a un intérêt pour l'étude de l'épilepsie, autant pour la détection, la localisation et le suivi des patients. Et puis, le développement du matériel et des méthodes d'analyse vont permettre éventuellement une intégration en milieu clinique. Alors voilà, j'aimerais remercier les organismes subventionnaires là, et tous les membres du labo, notamment là, Ali et Kay et Danem qui ont contribué aux résultats que je vous ai montrés.
Merci beaucoup, Dr. Nguyen, pour cette très, très belle conférence. Bien, s'il y a des questions, on aurait le temps pour une ou deux questions. Peut-être deux. Oui, Vincent.
Merci beaucoup, Dingue. C'est fascinant. Je voudrais revenir à quelque chose que tu as dit tout au début. Quelle est la base mécanistique du traitement cétogène pour l'épilepsie? Pourquoi?
Voilà, ça c'est des années et des années de recherche et des publications à en plus finir. Et la réponse simple, c'est qu'on ne le sait pas encore. Mais ce serait une réponse très simple. Là. Mais c'est sûr que ça joue sur plein de... Il y a de nombreux mécanismes qui sont impliqués. Mais à la base, c'est principalement au niveau de l'énergie, l'énergie des cellules, au niveau des mitochondries notamment.
Une question pour la précision de votre imagerie pour la concentration. C'est quoi la précision sur les cartes de concentration que vous avez? Donc, c'est en centimètres, c'est en millimètres? Ou...
J'entends pas.
Ah, désolé. Ouais. Donc, lorsque vous faites les cartes du cerveau avec les concentrations, c'est quoi la précision sur les cartes? Est-ce que c'est en centimètres ou c'est en millimètres? Ou...
Ah non, c'est sûr que c'est en centimètres. OK. Parce que c'est sûr que c'est moins précis que... la résolution est moins bonne qu'une la résonance magnétique fonctionnelle. La résonance magnétique fonctionnelle a aussi l'avantage de voir en profondeur. Nous, on voit seulement en surface. Heureusement, évidemment, l'épilepsie, c'est une maladie corticale du cortex. Ce n'est pas une maladie de la matière blanche ou des neuronucléaires centraux, ce qui nous permet que ce soit utile, évidemment, pour l'étude de l'épilepsie. L'avantage principal avec l'IRMF, c'est que l'IRMF, il faut que vous preniez un rendez-vous en radiologie. Vous avez 1 heure, puis c'est tout. Donc, les chances que d'enregistrer des pointes ou des crises, c'est relativement faible. Et puis, évidemment, vous ne pouvez pas surveiller le patient en continu. Donc, il y a des avantages et des désavantages pour chacune des techniques.
Puis, si j'ai du temps, j'aurais une autre question. C'est juste les détecteurs, si vous en placez plusieurs très proches l'un de l'autre, est-ce que vous pouvez utiliser l'information des différents détecteurs pour reconstruire une carte qui est plus précise ou qui est en profondeur?
Oui, c'est ce qu'on fait. Donc, l'important, c'est d'avoir la distance entre les émetteurs et les capteurs. C'est en général, on recommande entre 3 et 5 cm pour que les photons puissent aller assez profond pour toucher la surface corticale. Et puis, quand c'est trop rapproché, ce que vous allez avoir, c'est plutôt le signal qui provient des vaisseaux sanguins au niveau du scalp. Donc, ça, c'est des contaminants. Donc, il ne faut pas utiliser ce signal-là. Il faut d'ailleurs le soustraire. Donc, on va mettre des fois des capteurs plus rapprochés pour mesurer la contribution des vaisseaux sanguins au niveau du scalp, pour le soustraire de ce qu'on enregistre quand on prend le signal qui provient des émetteurs capteurs qui sont situés entre 3 et 5 cm de distance.
Merci.
Alors, bon, n'étant pas assez personnellement qualifié pour savoir dans quelle mesure ça a été couvert par votre réponse, je vais poser la question. Donc, Eric Samaru dit « Merci. Y a-t-il une limitation de la SPIRF sur la profondeur de l'enregistrement? Est-ce qu'une crise corticale profonde serait détectée? »
C'est ça. Donc, comme je dis, de l'émetteur, juste la lumière pénètre à peu près à 3 cm, mais si on soustrait le crâne, les méninges, etc., ça va pénétrer à peu près 1 à 3 mm au niveau du cortex. Donc, c'est sûr qu'on ne peut pas voir ce qui se passe en profondeur. Comme je vous dis, l'épilepsie étant une maladie principalement corticale des neurones, là, c'est moins un souci pour nous de ne pas pouvoir mesurer ce qui se passe en profondeur. Ouais.
Bien, merci beaucoup, Dr. Nguyen, et merci à la communauté pour les questions. Alors, now we will welcome my colleague Ifelowa Dedipe from the CRCHUM, Cardiometabolic Axis. She's PhD candidate at the laboratory of Dr. Ciaran Murphy-Royal. So, welcome to you and good luck.
Thank you so much for the introduction. I'll be doing my presentation in English. So I will be presenting on my talk titled Building Stress Resilience Through Astrocytes. This is just a brief overview of the work that I do in the Murphy-Royal lab, where we're looking at the role of astrocytes in the effects of early life stress on behavior. So to start off with, what is early life stress? It is any stressful experience that you experience during your childhood and formative years that has negative implications for our overall health and behavior. Although early life stress occurs in childhood, having an experience of early life stress actually has negative long-term implications for our health as adults, where an experience of early life stress results in an increased likelihood of experiencing psychiatric disorders such as major depressive disorder. So to summarize, if you're a child like Kevin from Home Alone and you've had a traumatic experience during your childhood, you have increased stress susceptibility and an increased likelihood of developing a psychiatric disorder compared to individuals who've had a relatively stress-free childhood, like myself. So in order to model this, we took a litter of a mother and her pups, and we separated the mother from her pups during their adolescence, as well as removed some of the bedding in the cage. After this, we then waited until the pups had reached adulthood, and then we looked at the long-term consequences of early life stress on cognition, as cognitive impairments are a pervasive symptom associated with psychiatric disorders. We did this by looking at threat detection, the ability to distinguish between a threatening tone and a neutral tone, by using a form of Pavlovian fear conditioning by presenting one tone with a series of foot shocks and another tone without. Thus, mice learn to associate one tone with a scary context and another tone without. Just as if you're on a hike and you hear a grizzly bear approaching, you know to run away because you have learned that any encounter with a grizzly bear could be potentially dangerous. So 24 hours after this, we wanted to assess how well these mice were able to distinguish between a threatening context, a threatening tone, and a neutral tone by presenting both of them independently in a separate context without foot shocks. So first, we wanted to establish that these mice actually learned that one tone was scary and another tone was not. So we looked at their freezing behavior. And mice freeze when they're faced with a threatening situation. And we saw that in both non-stressed and mice that experienced early life stress, both of these mice experienced higher freezing, as shown in the top part of both of these graphs, compared to the other tone, where they have less freezing at the bottom. Once we established this, we then looked at how does early life stress result in cognitive impairments in threat detection. And we found that mice that experienced early life stress experienced a significantly impaired ability to distinguish between the two different tones, meaning that they were unable to distinguish between which tone was scary and which tone was not. So does that mean that if you've experienced early life stress, that you are permanently predisposed to developing cognitive impairments associated with the psychiatric disorders? My goal here is to convince you that that is not the case. And our hope comes in the form of astrocytes. So as Dr. Murphy-Royal mentioned before, astrocytes are a non-neuronal cell involved in behavior, and these cells are actually stress sensitive. They have end processes that wrap around vasculature, which makes them the first point of contact for any changes in blood glucocorticoid or stress hormone levels. And actually, these cells express glucocorticoreceptors. And in situations of extreme stress, it has shown that these glucocorticoid-dependent activities result in astrocyte impairments in function and structure, thus having negative implications for behavior. So we wanted to ask, if we remove glucocorticoreceptors from astrocytes, what implications would this have for stress-induced cognitive impairments? So this is what we did. We took transgenic mouse lines and they underwent early life stress, and then we removed glucocorticoreceptors from astrocytes in the lateral amygdala, a region important for threat detection. So first we established, were these mice able to learn that one tone was scary and one tone was not? And we found that that was indeed the case. That, as you can see here in this figure, or in the graph, these mice experienced more freezing when presented with a scary tone at the top versus the neutral tone at the bottom, where they exhibited less freezing. We then assessed what effect does removing these glucocorticoreceptors then have on the ability to threat detect. And we found, even though both mice had undergone early life stress, when we removed the glucocorticoreceptors from the astrocytes, we were able to rescue the cognitive impairments induced by the stressful experience. So just to summarize, even if you're a kid like Kevin, our results show that a stressful experience that you experience early on in your life could potentially be rescued by targeting glucocorticoreceptors in astrocytes, improving or decreasing your likelihood of developing psychiatric disorders, giving you a similar likelihood to individuals who haven't had stressful experiences in their childhood. I'd just like to thank my wonderful lab, Dr. Murphy-Royal, and the wonderful lab mates in our lab and also the CRCHUM for this opportunity. I hope you enjoyed the talk.
Thanks a lot, Ife. It's always a pleasure to hear from your science.
Thank you.
No. And is there any question here in the room, or maybe? Okay. Wait. Okay. Yeah. Ah oui. Ah, c'est après. Ah, ok. On fait d'abord toutes les présentations. D'accord. Okay. We will take all questions at the end. We will first go with our second presenter, Shingo. Okay. Right? Okay.
Thank you.
Sorry for this. So now, please join me welcoming Shingo Nakajima, who is a postdoc in Dr. Stephanie Fulton Laboratory in the cardiometabolic axis. So, welcome here, Shingo. We are pleased to hear from you.
Thank you, Sabri, the kind of introduction me. So I'd like to talk in English about the role of fatty acid receptor in the protection for the neuroinflammation. The obesity is known the problem on energy metabolism. It also causes induce the inflammation in peripheral tissue like adipose and gut, and also causes in the inflammation in the brain. In the brain, as talked as Ciaran and Ifit, as astrocyte and glial cells is astrocyte and the microglia have respond to the immune function. These cells secrete cytokines and phagocytes of the injured synapses to regulate the neural activity. So it's known to the neuroinflammation as one of the causes of depression and anxiety. Nucleus accumbens is an important brain region that regulates the mood and motivation. So previously we found the high saturated fat diet that increases in the neuroinflammation in nucleus accumbens. The barrier inhibition of these inflammation rescued to the saturated fat diet induced anxiety-like behaviors. In nucleus accumbens, the medium spiny neurons are main cells. These cells respond to the glutamate and the dopamine input. So to test the effect of the neuroinflammation, we cultured the primary culture neurons from accumbens with the co-culture of the microglia by separating with the culture insert. We treated with lipopolysaccharide to induce the neuroinflammation, either the microglia or the neurons. After treatment of the lipopolysaccharide, the response to the dopamine and the glutamate is increasing in the nucleus accumbens neurons. Its effect was suppressed by the microglia, but the activated microglia treated with LPS, the light panel. Can I start? Okay, thanks. Also, the LPS treated the microglia, also the increases the information is the light showing the light panel, also increases the dopamine and the glutamate response in the nucleus accumbens neurons.
Attention, attention, code bleu annulé. Attention, attention, code bleu annulé.
Anyway, yeah, so the neuroinflammation is important to it's one of the factors to increase the neuron activity in the nucleus accumbens. So we focused the target to the N3 polyunsaturated fatty acid because it has the anxiolytic effect in hyperdiet-induced obese mice. Also, its receptor, the G protein coupled receptor 120, GP120 is known to regulate the peripheral inflammation through the beta-arrestin pathway. So interestingly, its receptor is highly expressed in the microglia. So we confirmed also using the microglia-specific YFP mice. So it's the YFP positive cells also highly expressed in the GP120 compared to the negative cells. So we tested whether the GPR120 activation in brain can rescue to the systemic inflammation, induce the abnormal behaviors by treatment of the LPS. The LPS treatment drastically induce the sickness behaviors and anxiety-like behaviors. Okay, so yeah, the CPD treatment is a good effect to rescue the systemic information by LPS that rescues both the sickness and the anxiety-like behaviors. So further, we tested the effect of the GPR120 activation in the nucleus accumbens-derived microglial cells. These cells are treated with a cytokine to activate the inflammation state. This treatment increases the cytokine production in nucleus accumbens microglia, the upper panel. Also, the picture showing the cytokine treatment, increasing the activated home, the microglia. But the CPD treatment rescue to the cytokine-induced cytokine production, and also changes also rescue to the morphological change by the cytokines. So it means that GPR120 agonist has a potential to regulate the microglia function. So the neuroinflammation is increasing the neuron activity in the nuclear subcameras. The GPR120 is a potential to regulate the microglia activity, which it means the GPR120 is a one novel target for the pharmacological treatment of depression and anxiety. I'd like to say thank you, my lab members, collaborators, and financial support. Thank you very much for your attention.
Thank you so much, Ingo, also for facing the challenge of presenting with two interruptions. Sorry for that. So yeah, we have time for one question, fortunately. So,
Shingo, great talk. I wanted to wonder if you could speculate on what you think microglia are doing. So, when you're getting this inflammation, you're getting this anxiety-like behavior. How are the microglia inducing this behavior?
Yeah, it's speculated the microglia circulated some cytokines. So I tested with using the co-culture model, which is separated. I think it's also RPS treated microglia, some circulated to some molecule to induce some molecules increasing the neuron activity. So I also treated with the cytokines to the nuclear subcameras neurons, also increasing the glutamate input. So I guess it's a cytokine, one of the components from the microglia. Thank you.
Ife, you have a question?
Oh, no.
No, okay, just took the microphone. Okay, so it's okay, if we can. Is there some question here? Not in the QR. I have maybe one very quick question to Ife. Did you, can you hear me? Yeah, okay.
Can I answer here? Is that okay?
Yeah. Did you evaluate the food behavior of mice subjected to childhood stress?
The food behavior?
Yeah, the food behavior.
No.
Okay.
We do measure like the weight of them before and after, but we didn't see changes. But I do have another lab mate that actually is looking at changes in feeding behavior due to changes in astrocyte activity and function.
Ah, ok, interesting to know. Yeah, thank you. So, merci à nos présentateurs pour cette deuxième section qui était consacrée à mettre de la lumière sur les neurosciences. Comme vous l'avez vu, c'était effectivement lumineux. Merci à eux tous.